Hello students, welcome to EPG Pathshala. I am Dr. Javed Ali from Department of Pharmaceutics, Faculty of Pharmacy, Jamia Ahmedabad, New Delhi. Today we are going to discuss about a module titled as Fundamentals of Cosmetics under paper cosmetics. Today we are going to study in that module Introduction to the Basic Thermodynamics as such. What is the theory behind formation of emulsion? The selection criteria for fragrance, stability and testing procedures, environmental safety, and industrial network. Introduction to basic thermodynamics, as the slide shows, the term thermodynamics was coined by William Thomson in year 1749, and it has been originated from a Greek word thermo, that means heat, and dynamics, that means power. The name heat and power is appropriate because thermodynamics explains the conversion of heat into power by steam engines. Thermodynamics in cosmetic reflects the stability and elegance of the formulation primarily. Thermodynamically stable cosmetic preparations, they show better stability in storage conditions. We are going to see the various basic laws of thermodynamics. The zeroth law of thermodynamics. In the case here, if two systems are in thermal equilibrium with the third one, they are also in thermal equilibrium with each other. Systems are assumed to be in thermal equilibrium with each other if spontaneous molecular thermal energy exchange between them do not lead to a net exchange of energy. This law is assumed in every measurement of temperature. Now coming to first law of thermodynamics, this is the fundamental laws of nature and is based on the principle of conversion of energy. The first law of thermodynamics states that during an interaction, energy can only change from one form to another form, and it can neither be created nor destroyed, but total amount of energy remains always constant. The second law of thermodynamics states that energy has quality as well as quantity, and actual processes occur in the direction of decreasing quality of energy. The thermodynamically stable cosmetic preparations are considered to be stable ones. There are various definitions of emulsions which are given. Now, in ancient times, there was a paradox as water and oil cannot mix. But the concept of emulsion demonstrated that these two phases, that is, the aqueous and oily, can be mixed very well to form stable formulation. It has been known for many centuries that emulsion was used for cosmetic purpose. Nowadays, there is an enormous scope of emulsion technology in cosmetics, toiletries, and healthcare markets. The reasons for popularity of emulsion include incorporation of both polar and non-polar materials in the same formulation. Earlier, emulsion technology has been associated with skincare products, but now the application of emulsion has become much wider. Currently, hair conditioners, antiperspirants, color cosmetics, and cleansing products are also based on latest emulsion technology. Emulsion can be defined as mixture of two or more immiscible liquids, or it can also be defined as a two-phase system which is consisting of two immiscible or partially miscible liquids, in which one is dispersed in another as very fine droplets. These two phases are oil and aqueous system, as already told. The term emulsion was coined from Latin word for to milk, as milk is a natural emulsion of fat and water with many more ingredients. Emulsion is mainly categorized into two categories, namely oil in water type and water in oil type. In oil in water type, in this the discrete droplets of oil are dispersed in water, and in water in oil type, in which discrete droplets of water are dispersed in oil. In addition to these two classes of simple emulsion, Some more complex systems are also there, such as three-phase complex emulsions, that is, oil in water in oil, which is represented as O oblique W oblique O, or water in oil in water, that is, water oblique oil oblique water. These newer classes offer greater degree of formulation flexibility with more efficacious and pleasant cosmetic products. As emulsions are thermodynamically unstable, the emulsifier is added to stabilize the emulsion system, and the emulsifier stabilizes the system by forming a thin film around the globules of dispersed phase. Of late, there are advanced classes of emulsion, namely microemulsion and nanoemulsion, with droplet size of less than 200 nanometer. Many theories have been given to explain the stabilization of emulsion by the emulsifier. At present, there is no single theory that can be applied universally to all emulsion. Rather, there are different theories, namely electric double layer theory, phase volume theory, hydration theory of emulsions, oriented wedge theory, adsorbed film and interfacial tension theory to justify the stabilization of emulsion via emulsifier. Now coming to electric double layer, which is also known as EDL or double layer, which is DL theory. The first theory for the electrical double layer was given by Goin and Champagne. DL is a structure which appears onto surface of an object when it is exposed to a fluid. It refers to the parallel layers of charge surrounding object. The first layer is of structure charge, may be positive or negative, comprising of ions absorbed onto the object due to chemical interactions. Second layer is made up of ions that are attracted to the surface charge via the Coulomb force. Theory of emulsions. The second layer is loosely associated with the object. It is made up of free ions that move in the fluid under the influence of electric attractions and thermal motions as well, rather than being firmly anchored. And it is therefore called as diffusive layer or diffusion layer. The magnitude of these forces increases with increase in the surface charge density as such. The theory of DLVO combines such double layer forces together with van der Waals forces in order to estimate the actual interactions potential between colloidal particles. The phase volume theory. If spheres of the same diameter are packed as close as possible, one sphere will touch 12 others, and the volume that spheres occupy is almost about 74% of the total volume. Thus, if the spheres of drops of the dispersed phase remain rigid, it is possible to disperse 74 parts of the dispersed phase in the continuous phase. But if the dispersed phase is increased to more than 74 parts of the total volume, a reversal of the emulsion will occur. However, the dispersed phase does not remain rigid in shape, but the drops flatten out when they come in contact with each other. Moreover, all the dispersed particles are not of the same size. Therefore, it is possible for the dispersed phase to be considered of 1 to 99% of emulsion. Now coming to hydration theory of emulsion, Fisher and Hooker stated that hydrated colloids make the best emulsifiers. Fisher stated that emulsifying agent by which a permanent emulsion is obtained invariably proves to be a hydrophilic colloid when water and oil emulsions are concerned. But another way, oil cannot permanently be beaten into water, but only into a colloid hydrate. Talks about the oriented wedge theory, and this theory has been given from the work of Langmuir and Harkins for the manner in which emulsions are stabilized. It is based upon the concept that molecules of emulsifier orient themselves at the interface between the dispersed and continuous phases, forming a wedge-like structure, the curvature of which determines the size of the dispersed phase. It explains the emulsion formation on the basis of selective solubility of surfactant. Surfactant possesses a polar and non-polar group, and they orient themselves in such a way that the polar group faces water and the non-polar group faces the oil. For example, as in case of soaps. Talks about the adsorbed film and interfacial tension theory. It is probably the most universally accepted theory for the formation of emulsions. This theory has been developed or rather extended from earlier theories which we have already gone through. And according to this theory, emulsification is influenced by the mass of the emulsifying agent. The ease with which this agent is adsorbed at the interfacial separating surface and the nature of the ions absorbed by the resultant film. Coming to the plastic film theory, which is also known as interfacial film theory, and according to this theory, the emulsifier is deposited upon the surface of the droplets of the dispersed phase in the form of plastic monomolecular film. This film prevents contact and coalescence of the dispersed liquid. Action of the emulsifier is purely mechanical and does not depend upon the surface tension. Now coming to the selection criteria and the selection type of different fragrance. Fragrance selection of a suitable fragrance or a perfume is a tedious task. A product which is intended to be applied to a person's body to make the person more attractive is defined as a cosmetic under the law. Some example of fragrance products that are regulated as cosmetics include perfumes, colognes, and aftershaves. Fragrance ingredients are also commonly used in other products such as shampoos, shower gels, shaving creams, and body lotions, etc. Even some products labeled unscented may contain fragrance ingredients which might react with active ingredients. This is because the manufacturer may add just enough fragrance to mask the unpleasant smell of other ingredients which have been used in the formulation without giving a product a noticeable scent. Some fragrance products that are applied to the body are intended for therapeutic uses, such as treating or preventing disease or affecting the structure or function of the body. Products intended for this type of use are treated as drugs under the law or sometimes as both cosmetics and drugs. Talks about the classification of fragrance, and we would like to classify fragrance. They are classified into various olfactory families or groups. The first group is the aromatic scents. It consists of fragrances from aromatic plants. They are widely used in aromatherapy and herbal medicine, and currently their popularity in high-end perfumery has been increased as well. These days, perfumes rarely belong to a single olfactory group. The second, aldehydic perfumes, they contain chemicals belonging to the aldehyde groups. And the aromatic perfumes are, these are made up of various herbs, rosemary, basil, or thyme. Example is gurelane. Aquatic perfumes is another class of perfumes, and they are a base of synthetic ingredients that evoke the sea, mountain, air, or fresh linen. And the example is IZ. The second class is citrus, fruity perfumes. This include grapefruit, lemon, mandarin, sweet orange, and fruits such as peach, red berries, watermelon, etc. The floral perfumes, they contain flower extracts such as rose, lily, or jasmine, and the example is pure poison. Gourmet perfumes, they contain vanilla and tonka bean, chocolate, or caramel. Example is Cartier and they consist of vanilla and tonka bean, chocolate, or caramel. Example is the Delices. The another class of fragrance is the green perfumes, which is formerly known as Kypre, are made of bergamot, patchouli, and oak moss. Example is Chanel. And the gourmand perfumes, they consist of vanilla, as already told. There may be oriental perfumes also. They have a base of amber, mass, vanilla, and other exotic plants. There may be woody perfumes. These are made up of woods such as cedar, sandalwood. An example is Mediterranean. Now coming to the stability testing of cosmetic. The prime purpose of stability testing of cosmetic product is to ensure that a new or a modified product meets the intended physical, chemical, and microbiological quality standards when they are stored under appropriate conditions. In other words, stability testing is an experiment in which samples of cosmetic preparation is put at different environmental conditions for a set period of time. And these conditions they vary in temperature, light levels, and meant to simulate what will happen to the product during its life cycle or shelf life. The present slide deals with stability testing of cosmetics, GMP, that is good manufacturing practices, described in 21 CFR Part 211, which is applicable to all over the counter OTC and prescription drug products can be applied to cosmetics. In some ways, and has been applied to nutritional supplement also. In case of stability testing of cosmetics, at selected intervals, evaluation of sample is required for various physical, chemical, and performance characteristics to see how they have changed. If the changes are found minimal according to standards, then it is said to have passed stability testing. This means that the cosmetic preparation has good stability and it will be stable for the duration of testing period. The underlying assumption in stability testing is that increasing storage temperature speeds up the aging reaction. A simple, handy thumb rule is that a sample stored at 45 degrees Celsius for 8 weeks is equivalent to one that is stored at room temperature for a year. This is not an exact predictor, but is good enough for the purpose of cosmetic products. Due to extensive variety of cosmetics and their inherent complexity, standard stability tests may not be prescribed. Manufacturers who have depth knowledge of their products and packages needs to modify testing protocols and to make a sound scientific basis to assess product stability. Specific tests should be performed in order to predict possible alteration in the product to be par with new unusual technologies and to be able to provide extended shelf lives to the product. New prototypes, whenever we make a new formula and are satisfied with the way it performs, we want to assess its stability. New raw materials, whenever we have to change the fragrance, color, or other raw material in a formula, we have to assess its stability to make sure there are not any unacceptable changes. Also, when we have a new raw material from a new source or a supplier, stability testing is required. New manufacturing procedure is focused on finding faster ways to make formulas. This often means to change some order of addition or shorten mixing time. Whenever changes like this happen, it could affect the formulation time. This further requires to run a stability test to see if the changes are acceptable. New packaging are when change in cosmetic products, they require diversified type of packaging and the material. Paper, glass, metal and plastic materials are generally used for cosmetic packaging. And the global market basically utilizes bottle, tube jars, containers, sticks, pumps, dispensers, roll balls, caps and closure, etc. for packaging purpose. Whenever the package is new, then it has to be checked that the formulation is compatible with container. Stability testing further ensure these parameters. How do we test stability of a cosmetic? There are no set rules on how to conduct a stability test for cosmetic products. Of course, for cosmetic OTC products, over-the-counter products like sunscreen or dandruff shampoos, FDA has specific stability test requirements that have to be followed. And there are some basic procedures which are described and can be followed for conducting a cosmetic formula stability test. A cosmetic preparation batch is designed and the amount of formulation is calculated based on the number of samples that will be used for the stability testing protocol. The glass jars are filled with the product along with the finished package, and both glass and packaging are subjected to testing if possible. So they are exposed to same type of environment, and the number of sample depends on how much testing is to be done. But at minimum, we should have 2 samples for each storage condition. Initial readings are noted, and once we have a sample filled, it is tested for all the characteristics we are going to evaluate later. The exact test depends on the particular product, but minimally physical characteristics such as appearance, color, and fragrances are recorded. The viscosity and pH measurements are also performed, and for aerosol products, spray patterns are tested. Samples are put in different conditions. Stability testing requires different temperature and light conditions. Some standard temperatures include 50 degrees Celsius, 45 degrees Celsius, 37 degrees Celsius, RT, which is the room temperature, generally recorded at 25 degrees Celsius and 4 degrees Celsius. A freeze-thaw stability test is also conducted, which involves cycling of product through 24 hours of freezing and then 24 hours of thawing, and different lighting conditions involving a fluorescent light box and a natural light box to simulate sunlight. Now, how we test stability of a cosmetic? The product evaluation includes samples should be evaluated at the following intervals of 2, 4, 8, 12, and 52 weeks. Only the room temperature, 37 degrees Celsius and 4 degrees Celsius samples are evaluated after 1 year. The highest temperature samples and light exposed samples need to be evaluated for the first 3 test intervals. The evaluation tests should be the same ones conducted when taking the initial readings. Lastly, stability is determined after 8 weeks. It is decided whether the formula is stable or not. Now, there are 3 different forms of stability tests. The first one is the physical and the chemical integrity test, which evaluate color, odor, fragrance, pH value, texture, viscosity, flow, and emulsion stability, because these are all signs of separation. Now, the various approaches which are used to predict how cosmetic will resist common stresses at higher temperature and light are generally part of physical and chemical stability tests. Generally, manufacturers determine whether to perform such testing based on the performance of the cosmetic product and its predictable shipping, storage display, and use condition. Common test procedures which are basically used for physical and chemical tests are like temperature variations. High temperature is commonly used as a predictor of long-term stability, whereas companies conduct their high temperature testing at 37 degrees Celsius and 47 degrees Celsius. If a product is stored at 45 degrees Celsius for 3 months, then it should be stable at room temperature for 2 years. Evidently, the product must be stored at 25 degrees Celsius for a period of 1 year. The second type of physical and chemical test is cycle test, and this tests include the product is passed through 3 cycles of temperature, testing from minus 10 degrees Celsius to 25 degrees Celsius. The product is placed at minus 10 degrees Celsius for 24 hours and the room temperature 25 degrees Celsius for 24 hours. This comprises of one complete cycle. If the product passes three such cycles, then it can be said that the product has a good degree of confidence in the stability. An even more rigorous test is a minus 10 degrees Celsius to 45 degrees Celsius 5 cycle test, and this puts emulsions under remarkable stress, and if it passes the test, it indicates that the emulsion exhibit very high stability. The third type, the centrifuge testing, is a dispersed phase of an oil in water emulsion which has a tendency to separate and rise to the top of the emulsion as oil droplets. This phenomenon is called creaming. Creaming is one of the first signs of impending emulsion instability, and this should be taken seriously. Centrifugation is a good test method to predict creaming of emulsion, wherein the emulsions are heated to 50 degrees Celsius and then centrifuged for 30 minutes at 3000 rpm. After centrifugation, the resultant product is inspected for creaming, and this test is necessary for the products which contain any kind of powders such as liquid or cream makeup. The next type of a physical chemical testing is a light exposure testing, in which both formulas and packaging can be sensitive to the UV radiation. All product must be placed in glass and the actual package in the window, and if it is available, a light box that has a broad spectrum output. Another glass jar totally covered with aluminum foil is placed in the window to serve as control. Frequently, we will see significant discoloration of the product and sometimes of the package too. This discoloration might be due to fragrance or some other sensitive ingredient. Usually all that is needed is the addition of a UV absorber, for example 0.1% benzophenol. Another type of physical chemical test is a mechanical shock testing. In order to determine shipping damage of the cosmetic and its package, mechanical shock testing is often conducted. Vibrational testing, for example, on a pallet shaker, may help in determination of demixing, that is, separation taking place of powders or granular products. Monitoring is done for all of the above mentioned tests. The color, odor, fragrance, viscosity, pH value, and if available, particle size uniformity and or particle agglomeration should be monitored under the microscope. The microbiological stability test, which is also a part of a stability testing method. In this, the microbial contaminants generally come from two different ways. Firstly, during production, and finally filling, and secondly, through the use of cosmetic by consumers. From initial, the cosmetic unit is opened by the consumer. A permanent microbial contamination to cosmetic is introduced due to contact with the consumer's hands and body. Microbial preservation is essential to make sure microbial safety of cosmetics for consumers, maintain the quality of the product, and confirms hygienic and high quality handling. Therefore, it is necessary to carry out routine microbiological analysis of each batch of the finished product. S. aureus, Staphylococcus aureus, C. albicans, Candida albicans, Pseudomonas species are considered as the main potential pathogens in cosmetic products. These specific potential pathogens must not be detectable in 0.1 gram to 0.1 ml of the cosmetic product. This parameter examines the criteria and method used, and the results obtained per batch should be well documented. Screening tests, these are the various simple testing kits available in the market, dip slides or plate counts, which provide quick and semi-quantitative results whether a cosmetic product is significantly contaminated or not. Sampling and evaluation of the results is simple and can be performed by personnel without any microbiological training. Another type of microbiological stability tests besides screening tests are the quantitative tests. Quantitative tests determine the actual count level of bacteria, mold, and yeast. These tests are very sophisticated and laborious and can be performed only by qualified personnel in professional microbiological testing laboratories. Methods of isolation for microorganisms from cosmetic products comprise direct colony counts and enrichment culturing. The third form, that is a packaging stability test, may directly affect finished product because of interaction which may occur between product, the packages, and the external surroundings. For example, product constituents may be absorbed into the container or may chemically react with the container. In addition, the container may not fully protect the product from adverse effects of atmospheric oxygen and/or moisture or volatile product constituent, for example, fragrances, which may evaporate through the container. Glass tests, in which the glass is the most inert material and widely used, which does not react with a cosmetic product in any way. For this reason, all testing should be done in glass and the actual packaging. In this way, it can be determined whether the cause of product failure is a formula or the package. Weight loss tests are also done in order to determine evaporation because water loss is through the container wall or closure gaps. Weight loss evaluation is one of the most important tests that must be conducted. This testing, which is performed in the actual package with a cap torque to 100% of the target torque, is done at room temperature and at 40 degrees Celsius for a period of 3 months. The weight loss should not exceed 1% per month for the package to be considered acceptable. Leaking test is also done on these packages. It is desirable to test the finished package product in different orientations, upright, inverted, or its side, etc., to determine whether the packaging may leak, especially during transport. Now coming to environmental safety and industrial network. The cosmetics we have seen, these are the products which are applied to human body for the purpose of cleansing, beautifying, and making the skin more attractive and pleasing. And the purpose of using cosmetic product is to maintain the body in a good condition, protect it from the effects of environment and aging process, alteration in appearance, and to improve the body smell. Cosmetic products are widely used for every socio-economic class of human beings, for cleansing, perfume, protection, and change in the appearance of the skin. Consumers' assumption is that the cosmetic products are safer and pose no risk to human health, but the actual scene is different. A number of complaints have been filed with various regulatory bodies which have found that sometimes the ingredients which are used to make formulations were not safe. A number of chemicals are usually added to cosmetic preparations as preservative and fragrances. Lots of them are found toxic and prohibited from usage as ingredient because they can cause cancer, mutation, reproductive toxicity, and endocrine disruption. Studies conducted by the Environmental Defense in Canada reveal that heavy metals such as lead, arsenic, antimony were found as ingredients which were not listed on the product label. These ingredients may be from raw materials or from processing methods. Similarly, high levels of mercury in cosmetic creams and endocrine disrupting chemicals were also found in cosmetic products. A study from Nigeria also indicated an elevated level of lead in facial talcum powders and other metals in personal care products. After these findings, many countries have taken steps towards regulation of cosmetic products, and a committee has been coined to see the cosmetic product for its toxicity. All this led to campaign for safe cosmetic. It was a result of sincere efforts of educational, religious, labor, and women's, environmental, and consumer groups with a goal to protect the health of consumer and workers. It propels the health and the beauty industry to make clear the use of chemicals which are linked to cancer, birth defects, and other health-related problems and substitute them with suitable alternative. It was started in 2002 with the release of a report, Not Too Pretty, Phthalates, Beauty Products, and the FDA. In this context, environmental and public health groups contacted with the laboratory to test 72 brand off-the-shelf beauty products for the purpose of phthalates, and the laboratory testing found in presence of phthalates in nearly three-fourths of the cosmetic product tested. Furthermore, the chemicals that were used in these formulations were not listed on any of the labels. A second report, Pretty Nasty, documented similar product test results in Europe. In February 2003, the European Union passed a new amendment to this cosmetic directive that prohibits the use of known or suspected carcinogens, mutagens, and reproductive toxin from cosmetic, which are together being termed as CMRs. This amendment came into force in September 2004, and in 2004, members of this Campaign for Safe Cosmetic and more than 50 other organizations signed a letter asking cosmetic companies and personal care product companies to sign the Campaign for Safe Cosmetic campaign for the global production of safer health and beauty products, a pledge to remove toxic chemicals and replace them with safe alternatives in every market they serve. The Safe Cosmetic and Personal Care Products Act of 2013. The purpose of this act was to ensure all personal care products should be free from harmful ingredients because existing laws, which were not being significantly updated since 1938, had many loopholes and allow use of chemicals linked to cancer, birth defects, disabilities, and other health-related problems. The cosmetic industry was facing serious problems because beauty products, shampoos, and lotions, etc., were contaminated with toxic chemicals like cancer-causing formaldehyde in baby shampoos, lead in lipsticks, and mercury in skin creams. The Safe Cosmetic and Personal Care Products Act of 2013 gave the beauty industry a much-needed makeover. This act proposed to substitute ingredients that are linked to cancer, birth defects, and other developmental harm to make health-based safety standards. Those are used for protection for children, elderly workers, and other segment of the populations. To close labeling loopholes by providing full ingredient list on labels and company website also. In addition, to provide access to information about unsafe ingredients in cosmetics, to share the data for avoidance of duplicative testing and promote the other alternatives to animal testing, to provide sufficient funding for FDA Office of Cosmetics and Color so that it has a resource that report error of the cosmetic industry. Now coming to industrial network, voluntary self-regulation by the US cosmetic industry is considered by many to be both a regulatory anomaly and an excellent success. No industry in the history of US has ever made a greater commitment to self-regulation or has been more successful in achieving it than the US cosmetic industry. Virtually all aspects of cosmetic industry are subject to some form of voluntary cosmetic toiletries and fragrance association, which is also known as CTFA program, initiated by the industry itself to assure FDA, the medical profession, and the public that cosmetic products are safe and appropriately labeled. The cosmetic industry is sensitive to the image of an uncontrolled market where anything goes. Protecting industry independence entails ensuring adequate industry safety levels in order to avoid the need for increased federal regulation. It would externally impose more stringent requirements upon the industry. Thus, the cosmetic industry since the 1960s or so has relied on self-regulation, particularly in times of problem, to convince Congress not to enact new, more intrusive legislation. And here now coming to summarization what we have learned in this module, we have talked about basic thermodynamics laws, we have talked about the theory of emulsification and the related physical stability issues which are interrelated with these theories of emulsification. We have gone through a protocol how fragrance are selected. We have talked about the stability testing protocol and safety and industrial network.